Alright, hello everyone. How's it going? Mr. Binder here. This is the second screencast I'm going to do for you guys. The first one that we took a look at, it was functional groups, and there was a couple of them that I wanted you guys to identify, mostly on the macromolecules that we're going to be taking a look at in this section here. Now remember, macromolecules are the building blocks of our cells. We're made up of cells. Our cells are made up of macromolecules. Okay, remember, there's four of them that we're going to talk about: carbohydrates, proteins, lipids, and nucleic acids. Okay, we're going to go through each one. We're going to talk about their structure, some things that you guys can do to indicate whether it is a carbohydrate, a protein, a lipid, or a nucleic acid. And then we're going to go over the subunits that each of these macromolecules are made up of. So if you take a look at the picture here to the right, you see these different pictures of molecules here. We see the blue, the green, okay, and the orange there. Notice how proteins, polysaccharides, which is another name for carbohydrates, and the nucleic acids here, they're all made up of what's known as subunits. So we're going to cover what each of these macromolecules are made up of in terms of a subunit. Now all these macromolecules are organic compounds, so that means that they contain both hydrogen and carbon. The first one that I want to talk about is carbohydrates. These are basically your sugars. And we're going to go over a couple different terms that you guys might not have heard of before: disaccharides, monosaccharides, and polysaccharides. They're all examples of carbohydrates. Okay, complex carbohydrates are more of like your polysaccharides. All right. Now taking a look at your carbohydrates here, basically they're made up of carbon, hydrogen, and oxygen. That's it. The general molecular formula here is CnH2nOn. And what does that mean? Well, if this molecule here, or whatever molecule we're taking a look at, has 6 carbons, well, it has to have 6 oxygens. But because there's a 2n there for hydrogen, it has double the amount of hydrogen, meaning that there's 12 hydrogen. Its roles in cells, both in plant and animal, is for energy storage and obviously structural support in cells and tissues. All right, this molecule is hydrophilic. All right, and essentially when we take a look at the subunit of carbohydrates, or these complex carbohydrates, what we're going to find out is that they're made up of rings. Okay, pentose and hexose ring structures. All right, pentose meaning that there's 5 carbons in a ring structure, or hexose meaning that there's 6 carbons in a ring structure. So the subunit of carbohydrates is called monosaccharides. This is the simplest type of a carbohydrate. Okay, essentially what our cells use monosaccharides for is to make ATP. It's very, very important for our cells to use this to make ATP, because if we don't, then obviously our cells will not function properly. So what are some names of these monosaccharides that our cells are going to be using to make ATP? Well, in chapter 4 we're going to take a look at glucose. Okay, this is found in mostly all of the sugars that we are going to be eating. Again, fructose here with honey, fruit, and juices. Okay? The most common monosaccharides that we're going to be taking a look at here is probably the 6 carbon. That would be your glucose molecule. That's a hexose ring. Okay, there are other monosaccharide ring structures like pentose, okay, that we will take a look at, but let's stick more towards the glucose molecule. There are 3 carbon linear structures. The reason why they are their ring structure, okay, and basically just because there's a reaction between the linear molecule here. If you take a look at the picture to the right, you'll see that there is a carbonyl group at the end of this linear structure here, and it's going to react with one of the hydroxyl groups of the same molecule. That will form your ether here, creating this nice ring structure. Okay, and essentially what happens here is it creates a 6 carbon ring structure. So 1, 2, 3, 4, 5, and 6. Okay, I want you guys to always label the ring structure like that, only because when you do, let's say, try to identify a reaction, or maybe I'll have you guys draw a reaction on the test, I want you guys to start with number 1, because this is where the actual linkage between glucose molecules is going to occur. Okay. Glucose, remember, this is the most important monosaccharide that we are going to be talking about. Okay, taking a look at glucose here, there obviously are linear and ring structures, but mostly the naturally occurring structure that glucose will be talking about is the ring structure. Okay? There's 2 possible arrangements, alpha glucose and beta glucose. The difference between the 2 is the position of the hydroxyl group, okay, functional group, on the number 1 carbon. Notice how they are numbered here for you. Okay, so if it is, I like to call it, if the hydroxyl group is positioned in the down position, it's called alpha glucose. If it is positioned in the up position, it's called beta glucose. And when these 2 molecules are different, and the reason why they're different is because they are structurally, well, from a same molecular formula, they're the same, okay? But structurally, they're different. And that makes their molecule structure totally different from each other when they are linked together, and we'll take a look at that. All right, alpha glucose. If you link alpha glucoses together, essentially you're going to get glycogen. Or what you could do, alpha glucose is the subunit of amylose, okay, which is essentially just starch sugar. Now, long chains of beta glucose, that forms the actual what's known as cellulose, and that's found in plants, and we can't digest cellulose, so thankfully there is an isomer of beta glucose known as alpha glucose, as if there wasn't, then we wouldn't be able to digest it, and then we'd have to have some other form of energy that our cells would have to use. Now disaccharides, obviously, they're naturally occurring as well. Now a disaccharide, some examples here, sucrose, lactose, and maltose. Notice the makeup of each of these disaccharides. Okay, they're made up of 2 monosaccharide units. Okay, glucose, fructose, galactose, glucose, and glucose and glucose. All right. The examples I give you here: maltose, used to make beer. Okay, sucrose is a simple sugar. You use that in your coffee if you do drink coffee. Lactose, obviously, in your milk, and we need to digest lactose. And sometimes some of us might be lactose intolerant, meaning that we can't digest that, and that creates issues. So, but taking a look here, disaccharides, all right, they're formed, all right, in our cells, but they're also broken down in our cells. If we were to take a look at the reaction between 2 monosaccharide units here, okay, we have 1 and we have 2. Taking a look at these 2 monosaccharides and having them form together, notice the 1 carbon of one of the glucose molecules serves as the reaction center. Okay, that's very important. Now, here, the reaction is taking place between the number 1 carbon of the first glucose molecule and the number 4 carbon of the second glucose molecule. Notice the reaction that will bind these 2 glucose molecules together is known as a dehydration synthesis reaction or condensation reaction, only because a water will be formed as a product when these 2 glucose molecules are combined. Notice when we take a look here, this is a 1,4 linkage, okay, because it's linked to the 1 carbon of one glucose and the 4 carbon of another glucose molecule. Some common linkages, and this will be a multiple choice question: 1,4, 1,2, 1,3, and 1,6 linkages. Okay, the bond that forms between the 2 glucose molecules is called a glycosidic bond. Okay, remember that because that's also a multiple choice question. Complex carbohydrates or polymers. Okay, you're going to see all of these terms. Don't get confused, they're all the same. Okay, complex carbohydrates are polysaccharides. Okay, polysaccharides are polymers. Polymers are essentially long chains of the same unit, okay, attached together. And the examples that I gave you were cellulose in plants, okay, and amylose, which is something that we digest and we need in order to make ATP. Now there's two different types of complex carbohydrates. There's unbranched, okay, and branched. Now the difference between the two, okay, is obviously their structure. When you take a look at starch, notice it's kind of helical in structure, but these molecules are attached together, probably the same linkage, all right. When you take a look at cellulose here, all right, it's very linear in structure, okay. And then the other thing is unbranched. When our cells, let's say we just ate a huge meal, we digested all of the sugars, and we don't need it in our blood anymore, so our cells are going to store it. Our cells store it as glycogen. Now take a look, that glycogen here is branched. And the difference between the two, okay, is that there are different types of linkages here. So we might see a 1,4 linkage there, but see how there's a branch there. So we might see, maybe a 1,6 linkage there. Okay, keeping that in mind, that glycogen is definitely branched. Okay. Polysaccharides, they're assembled by obviously a dehydration reaction. Okay, the bond that forms in between the two, or all this whole molecule, is known as a glycosidic linkage. Okay, that's found here. All right. Essentially, polymerization is just the term to link identical, okay, glucose molecules together. All right. Linear unbranched hydrogen bonds form between the molecules. All right, and that goes back talking about here. Okay. Essentially, what happens here is that this molecule, okay, this hydroxyl group will form a hydrogen bond between another strand or polysaccharide. Right, polysaccharides are polar. That means that they share a charge across the molecule. Okay, it's positive to negative. They are hydrophilic, meaning that these are water-loving, okay, molecules, but they're non-soluble in water. The reason why they're non-soluble in water is because the bigger the molecule gets, the more insoluble in water it becomes. Again, some examples. The amylose, that's the starch that we eat, made up of alpha glucose. Okay? Glycogen, we store extra alpha glucose molecules in our cells as glycogen. Okay? Cellulose is made up of beta glucose molecules, and plants use that for cell walls and essentially for structure and support. And this last one here, this chitin or chitin, however you want to pronounce it, this is the exoskeleton of insects and crustaceans. All right, that's essentially carbohydrates. The next section I want to talk about is lipids. Now, lipids, very important molecule. All right, essentially, we're going to take a look at what makes up, or the derivative of, most of these lipids. Okay, they're a little bit different from carbohydrates only because they're not as large as carbohydrates are. Okay, and they also have some different properties. Now, lipids are nonpolar. Okay, that means that there isn't a difference in charge across the molecule. They're mostly made up of carbon and hydrogen. Okay, they are not polymers. That means they are different from carbohydrates. Now, because they are nonpolar, water's polar, lipids are nonpolar, they do not dissolve in water. That's very, very important here. Some of the roles of lipids: formation of cell membranes, okay, energy source, hormones, and vitamins. Okay. There's 5 categories that we're going to talk about: fatty acids, fats, phospholipids, steroids, and waxes. This is a multiple choice question. Fatty acids are the derivative of most lipids. Okay, they provide the structural backbone of most of these lipids. All right, taking a look at the picture, what we see here is just long chains of carbon and hydrogen. Okay? Every time you see a peak, that's a carbon. Every time you see a little peak here, that's a carbon. Okay? And so on and so on. Those are all carbons attached together. Remember, the term is catenation. Now, taking a look at each of these peaks here, we see hydrogen. Okay? Now, what I want you guys to focus on, okay, you see these little double lines here. Well, whenever you see that, that's a double bond between the carbons. All right. Essentially, another thing to note here or take a look at is the functional group that's found on fatty acid chains. Okay? And that's a carboxyl group. This carboxyl group is very important in terms of forming triglycerides and your other larger molecules. Now, this is what gives this fatty acid its acidic properties. Again, this is going to be a multiple choice question. Okay? And then again, as the chain length increases, insolubility in water increases. So that means that we're going to add more carbons to the chain. Okay? Two different types of fatty acids, saturated and unsaturated. Again, what that means is if a fatty acid is saturated, notice over here, there's no double bonds in the chain. Okay, they're all single bonds. That means that each carbon has the maximum number of hydrogen molecule or hydrogen atoms that can be attached to that carbon. Okay, and unsaturated, that means that there's a double bond that has formed in between the carbon molecules. This doesn't allow for the maximum number of hydrogen atoms to attach to the carbon molecule. All right, and there's a couple different types of unsaturated. Mono, meaning there's one double bond in the chain. So this here would be an example of a monounsaturated fatty acid. Okay? And then there's polyunsaturated. That means that there's more than one double bond. What happens in these fatty acids is it causes a bent shape. Okay? The difference between the two is saturated fatty acids are more solid at room temperature, so that would be more like your butters, okay, or your margarines. Unsaturated fatty acids are like liquid at room temperature. An example would be like your oils, okay, that you might use for cooking. I do want to talk about cis and trans fats. Okay, the difference between the two, okay, they both have a double bond in the chain here. The difference between the two is that cis is naturally occurring, okay, good for your health, and it forms kinks in the carbon chain. Now, the reason why it forms kinks in the carbon chain here is when you take a look at the two hydrogen molecules that are attached to the carbon, double bonded to carbon, they're on the same side. All right, when they are on the same side like that, that will form the kink. Okay, they're naturally occurring, and they obviously these fatty acids will help to form our cell membrane, okay, or plasma membrane. Trans fats are artificially produced, and what trans means is across. So the hydrogen atoms are across from each other when there is a double bond between the carbons. Now, this is bad because they're artificially made. They're found in a lot of processed foods, and they're bad because they contribute to heart disease. All right, they raise your bad cholesterol or your LDL. Okay, and they like to build up in the arteries of your or in the walls of your arteries, and it could definitely cause some issues down the road in terms of heart disease. All right, so you really want to be careful about that McDonald's. Next section, fats. Okay, what is a fat? Well, it consists of a couple things here. Okay, a glycerol molecule and 1 to 3 fatty acid chains. So we took a look at fatty acid chains. All right, this right here. Now what we're going to do is take a look at multiple fatty acid chains attached to a glycerol molecule. Okay, now notice the different circles that are being drawn there. That's where the reaction will occur between the glycerol and the fatty acid. Okay, it's between the carboxyl group of the fatty acid chain and between the hydroxyl group of the glycerol molecule. These fats are formed again by what's known as a dehydration synthesis reaction. Okay? The fatty acid chains can be the same fatty acid chain or could be different. okay, depending on what's needed in the cell. Right? These molecules are hydrophobic. That means that they are not, or they're afraid of water. Okay, so they do not like water at all. All right, and the formation or the bond that's the bond that's known here between the fatty acid and the glycerol molecule. This is known as an ester linkage. Okay, this forms between the fatty acid chain and the glycerol molecule. Essentially, the most well-known fat, triglycerides. Tri meaning there's 3 fatty acid chains attached to a glycerol molecule, and you can see the reaction here between the hydroxyl group of a glycerol and the carboxyl group of a fatty acid. Okay, the dehydration synthesis reaction occurs, we have your triglyceride. All right, triglycerides are found in our food. We eat food, our body uses these triglycerides, okay, as an energy source, as well as, glucose. But what triglycerides do is if we eat too much of them, then we store them. We store them in our fat cells or adipose, okay, cells. And what could happen, obviously, is we get those love handles, you get that little belly there, because we're eating foods that are high in triglycerides. All right, liver, it will produce triglycerides for extra energy. Okay. The cool thing about triglycerides here is that it does contain more energy than carbohydrates. But we will only be taking a look at glucose for cellular respiration. Phospholipids. Okay, phospholipids. Taking a look at phospholipid, what do you notice about the fatty acid chains? There's only 2 now instead of 3. Okay, on one of the hydroxyl groups of the glycerol molecule, we see a phosphate group and a polar unit. Okay, this is obviously different from triglycerides. Our phospholipids, they are the main component of the plasma membrane. All right, and they're cool because they are known as amphipathic molecules. Amphipathic meaning that they have both a polar part, okay, and a nonpolar region. Okay, meaning that this molecule loves water at one end and hates water on the other end. And this is why our cells are made up of a bimembrane, okay, a phospholipid membrane, or bilayer. Okay, phospholipids, obviously their role here. Taking a look, we've got the polar region, we've got the 2 fatty acid chains. Okay, the bilayer forms naturally because we're made mostly of water. So the hydrophilic heads will be exposed to the water. The hydrophobic tails won't be exposed to the water. Okay. When you take a look at the fatty acid structure here, notice the kink. See how there's some extra space there? These unsaturated tails will make the membrane more permeable to water and small molecules. And remember, we discussed fluidity. All right, the fatty acid composition here can change over varying temperatures, allowing for more molecules to cross the membrane when needed. Steroids. Okay, these aren't the ones that we take before we go to the gym to get big muscles, right? Essentially, our body makes steroids. Okay, and we're going to talk about a couple different structures here. Notice when you see a molecule and it has 4 fused carbon rings, okay, this is an example of a steroid. Okay, all right, a couple things that we're going to look at here. Okay? This steroid molecule consists of a hydroxyl group. Okay, they're nonpolar. All right, and they are amphipathic molecules, meaning that they share both hydrophilic and hydrophobic properties. Some examples here are cholesterol. Okay, that's found obviously in our body, and phytosterols, those are found in plants. Cholesterol, okay, this is formed in the liver. Notice the 4 fused carbon rings in cholesterol. So cholesterol is a steroid. All right, 4 fused carbon rings there, formed in the liver. We also get it from our diet as well. Okay, and there's 2 different types, HDL and LDL. Okay, and we'll talk about that a little bit later on. Well, we already know about cholesterol because it's the structural component of the plasma membrane. Notice how it embeds itself in the membrane here, and it helps to maintain that fluidity of the cell membrane over varying temperatures. Very important. Remember, cholesterol is amphipathic. Okay, some of the functions again. Okay, and maintains the integrity of the membrane. Okay, proper membrane permeability, fluidity, fatty acid transport, metabolism across the membrane. Okay, and cholesterol will be used to form some of the hormones, bile salts, and other vitamins in the body. Now remember, cholesterol cannot dissolve in the blood. Okay, so it's transported by carriers, and what these carriers are, they're protein carriers. Okay, because proteins can dissolve in the blood here. So there's two different types, LDL and HDL. We get a lot of this stuff from our foods that we eat. So taking a look here, a big burger, we eat it, we digest it, we absorb it through the intestines into our blood vessels, and it's floating around our blood vessels. Well, in excess here, what will happen is the triglycerides and the LDL, okay, that we get from our diet will build up in the blood vessels and could definitely promote cardiovascular disease. Now, our liver does make HDL, or high density lipoprotein. Okay, and essentially what this does is it removes the LDL, okay, from the arteries, brings it to the liver for destruction, okay, or for removal. Now, what's cool about this is that HDL, okay, is made by the liver, okay, and high levels of high density lipoprotein, or HDL, will not promote cardiovascular disease, but low levels of HDL will promote cardiovascular disease. So you really got to keep this in balance here. There's actually a study that was done, maybe a year or two ago, and taking a look at cholesterol, and now that it's maybe not the molecule that scientists or should be blaming for heart disease. Right? They did a study in people who were a little bit older, probably in their 60s, who had high levels of cholesterol, and it didn't show any signs of heart disease at all. So there's there's some journals out there that are obviously a little contradictory of a lot of different things, but for what we need to know now, there's two different types of cholesterol. One's LDL, the other is HDL. One promotes cardiovascular disease and the other doesn't. Sex hormones. Okay, notice the 4-fused carbon ring here. Okay, we'll take a look at these more when we discuss homeostasis. Okay, but essentially here is, sometimes you can just tell, right? Testosterone produced by males, estrogen produced by females, okay, progesterone obviously produced by females as well. This is what develops our sexual traits in our sex cells, and we'll discuss that a little bit later. We don't need to know any of these. These are the harmful effects of steroids, obviously. Hopefully you guys don't take them. I don't see anybody in the class that would be taking them, so just review these harmful effects of steroids. The last example would be our waxes. This consists of long fatty acid chains, okay, with an alcohol molecule or carbon ring. I don't really ask you to know too much about waxes. Just know that they're nonpolar. They're kind of soft solids. Okay? And some of the functions, this is something that I might ask you in a multiple choice, is that it serves as waterproofing for plants and animals. All right, cutin is the plant's protective waxy coating on the leaves. Okay, if you take a look here, it repels the water from the leaves. And beeswax, production of honey. I believe the wax that our bodies produce is called sebum. Okay, and it's actually a good defender against pathogens like bacteria or viruses. All right, that's essentially it for lipids. We covered carbohydrates, lipids. Now what we're going to do is get into proteins and nucleic acids. All right, taking a look at proteins here. All right, it's a polymer, so it's linked. Okay, made of many subunits. Okay, and what is that subunit? Well, the amino acid. Okay, long chains of amino acids attached together. Essentially, what's going to happen here is because of these amino acids attaching together, they are going to fold into different 3D structures. All right, so we're going to talk about each of them: primary, secondary, tertiary, and quaternary. Right? There is a difference between all four of them, and I will definitely ask you this question on the test. Okay? The folding of these amino acid chains will allow for the protein to function. Okay? Again, the reaction here that we see that forms between the amino acids is called a dehydration reaction. Okay? And that's going to occur here between the amino group of one amino acid and the carboxyl group of another amino acid. Okay? That's known as a peptide bond. All right, so the ester linkage in lipids, glycosidic linkage or glycosidic bond in carbohydrates, and proteins, the bond that links two amino acids together is known as a peptide bond. There's 20 different amino acids. This number is essentially changing all the time. 8 essential. Okay, there is 9 that are essential now, which needs to be supplied by the diet. That means if there's 9 essential, there's 11 non-essential amino acids. That means that our cells can make those amino acids. We don't depend on our diet to obtain those amino acids. One thing I do want you to take a look at, amino acids, this is the skeletal structure here. Okay, we see an amino group attached to a central carbon as well as a carboxyl group. This is the central carbon here. Okay, this is your skeleton structure of an amino acid. The difference between the 20 is this R group here. This R group, there's 20 different R groups. Okay, and they could be as simple as a hydrogen to as complex as, let's say, a ring structure. Right? Do I want you to know all the amino acids? No, I don't. What I do want you to know is the skeletal structure. Okay. These are the 20 different amino acids, and look, each side chain or R group here. Okay, that's the highlighted part here. Okay. That's what will allow this amino acid different properties. So we see some nonpolar amino acids. Over here, we see uncharged polar amino acids. Okay? Then we have polar amino acids, negatively charged, and positively charged polar amino acids. Okay? I don't want you to remember each of these. Okay, we'll take a look at these amino acids a little bit later. Okay, what I want you guys to do is just understand the skeletal structure of an amino acid. All right, it's that amino group, that carboxyl group. They're both attached to a central carbon, okay, as well as an R group, and that R group can be one of 20. All right, taking a look here, okay, the bond occurs between the carboxyl group of one amino acid and the amino group of another. Remember, dehydration synthesis reaction forms this peptide bond here. So dehydration means the removal of a water molecule. Right? Taking a look at a string of peptides, we see about 1 to 49 amino acids. It's known as a peptide. A polypeptide is a string of 50 or more amino acids. Okay. Here's some examples of proteins, okay, and how our body uses these proteins. Essentially for structure, all right, so like hair, tendon, or ligaments, all right. Proteins are used in as a defense, basically antibodies, all right. That's over here, this little Y structure here. These antibodies are signals, okay, that will identify pathogen. So like bacteria or viruses that are floating around in our bloodstream. And again, when we get sick, we develop these antibodies, okay, and what happens when that pathogen, again, makes its way into our body, these antibodies are ready to identify it. That way our body can take care of it a lot quicker. Proteins are also hormones. This will be discussed more when we do talk about homeostasis. Transport across transport of materials, again, hemoglobin. Hemoglobin is very, very important. Okay, it is a protein that's found in our blood or our red blood cells, and it's used to bind oxygen when we breathe it in. Again, recognition and receptors Okay, the histocompatibility complex, you don't need to know about that, but cellular markers are signaling other cells using this histocompatibility complex. Enzymes are important. Okay, the reason why enzymes are important is because they're catalysts. And again, for mobility, the movement of these actin and myosin proteins, all right, that's this picture down here. That's essentially important for muscle contraction. Now let's take a look at the different protein structures. There's 4 of them: primary, secondary, tertiary, and quaternary. All right, we'll go through each of them. Primary is essentially just a linear sequence of amino acids. Okay, that's it. So amino acids attached together, forming a long chain. Okay. The secondary structure here is a little bit different, okay, only because the difference between a primary and a secondary structure is the interaction of these R groups. Okay, the interaction of these R groups will allow this protein to kind of fold, all right, and form more of a different shape. And the result of this secondary structures is from hydrogen bonding. Taking a look here, the hydrogen bonding occurs between different amino acids. And a hydrogen bond is a bond between a hydrogen and an oxygen here in this case. It could also be between a hydrogen and a nitrogen. Okay, it's just hydrogen bonding. Now it's not a physical bond. Okay, it's just an attraction between atoms. Okay? In this case, a hydrogen and an oxygen. So this is not an actual sharing of electrons. This is just a force that's bringing these two atoms together. All right, it's probably because difference of charge across the molecule. Take a look here. Secondary structure: beta-pleated sheets, okay, and alpha helix. Okay, taking a look at the some examples. Silk, okay, is obviously an example of beta-pleated sheets. Your filamentous proteins or your transmembrane proteins, those are your secondary structures, and they are found in the cell. Tertiary structure. This is now we're getting into a 3D shape. Okay, this is a polypeptide chain that has a 3D shape. Now some of the intermolecular forces, okay, or intermolecular reactions that are occurring between the polypeptide, there's a couple, and these are responsible for the 3D shape that takes place here in this tertiary structure. Notice hydrogen bonding. We've already talked about that. That's the only intermolecular force we see in the secondary structure of proteins. There's 3 more in tertiary structure that will allow for this 3D shape. Hydrophobic interactions, okay, disulfide bridges, and ionic bonding occurring. All of these intermolecular forces promote this 3D shape. Okay. Now, this is something that I will probably ask you on the test. I would want you to identify the different intermolecular forces and label them. So remember this picture. The last protein structure we're going to talk about, okay, is quaternary. And what's the difference between tertiary and quaternary? Well, we see the interaction of multiple polypeptides. Okay? Tertiary, there was only one polypeptide, and it had a 3D shape. Well, quaternary, we see multiple polypeptide chains, okay, more than one. Still, same intermolecular forces that are found in tertiary, but here we see the interaction of multiple polypeptides together. Okay. Essentially, what I want to talk about, okay, protein prosthetic groups. In order for a protein to function, it needs a non-protein component, and we'll talk more about this when we discuss enzymes. But the example I give you is hemoglobin. Okay, and this is a huge question on the test, and I want you to know this for the test. Okay, hemoglobin is quaternary in structure. Notice it's quaternary because it's made up of 4 different polypeptide chains. Okay, I'm not going to circle the other 2, but when you take a look at that, okay, that you can tell that that's quaternary in structure. Again, hemoglobin is very important. The reason being is hemoglobin is the protein that will bind oxygen and carry it around to our cells that need it. Okay? Now, in order for hemoglobin to function, it obviously needs a metal ion. And these are known as prosthetic groups. Okay? And there's multiple prosthetic groups. Okay? Cofactors and coenzymes, those are things that we'll be discussing when we talk about enzymes. For this part here, for hemoglobin, it's not an enzyme. It's just a functioning protein that will bind oxygen. Okay? And it needs a couple different things. Notice the heme molecule is one of them. Okay, let me get a different color here. The heme molecule is one of them. All right? And the heme molecule here, okay, it's a porphyrin ring. Okay, almost kind of looks like a dream catcher. But what do you notice in the middle of this porphyrin ring? Well, it's iron. So not only does hemoglobin need this heme molecule, it also needs iron. And this is exactly where oxygen will be captured. Okay, so know these 4 different points here. All right. Last question though, how many molecules of oxygen can hemoglobin carry at one time? Well, if there's 4 polypeptide chains, 4 heme groups, 4 iron atoms, well then 1 hemoglobin molecule can carry 4 oxygen molecules at one time. Now, how many hemoglobin molecules are there per red blood cell? Well, there's anywhere from 200 to 250 million hemoglobin molecules per red blood cell. That's crazy, because if you do the math, each red blood cell can carry about 1 billion oxygen atoms at a time. What's going to happen here, okay? Protein denaturation. Essentially what happens is when proteins are exposed to differences of pH or maybe temperature, high temperature ranges, okay, it could start to denature. What that means is that proteins can start to lose their function. All right. When this occurs, it's often irreversible. Okay? And again, it happens over extreme temperatures and pH, and obviously with the use of chemicals. All right, the last macromolecule we're taking a look at. All right, remember we took a look at carbohydrates, lipids, proteins, and again now nucleic acids. All right, this is our last group. Nucleic acids, the 2 molecules we will be taking a look at here, DNA and RNA. Okay, I will be asking you a question on the test to compare and contrast DNA to RNA, and there are obviously some differences. Taking a look here, RNA is single-stranded. Okay, single-stranded molecule. Okay, DNA is double-stranded. Notice the 2 strands connected together. Okay, remember that. Another difference is the nitrogenous bases that are made up of both DNA and RNA. Take a look here. C, G, A, and T are made up of DNA. Okay. Now, what makes up an RNA molecule? Well, we have C, G, A, but instead of T, we see a U here. Okay, and that's going to be very important when we discuss protein synthesis. Okay. The subunit of nucleic acids is known as nucleotides, and the link between these nucleotides is a phosphodiester bond. What does nucleotides consist of? Well, a nitrogenous base, a sugar, and a phosphate group. Now, taking a look here, okay, a nucleotide essentially is 1 phosphate group. Okay? A nucleoside diphosphate, that's made up of 2 phosphates. A nucleoside triphosphate, right, is made up of 3 phosphate groups. Essentially, when our cells build DNA, okay, through the process of DNA replication, it will use these nucleoside triphosphates, okay, to build the DNA. All right, but taking a look at the functions of some nucleotides, right? For data storage, obviously DNA, this is our most essential molecule in the cell, is because it has all of the instructions for our cell. All right, ATP, all right, is used as energy currency in the cell. Some cellular communication with CAMP and ATP molecules, as well as coenzyme catalysis. Taking a look at the nitrogenous bases here, this is going to be a multiple choice question. Pyrimidines, okay, uracil, thymine, and cytosine, and then your purines, your adenine, your guanine. The difference between the two is there's 2 carbon rings as opposed to just one. Okay. So when I ask you a multiple choice question, I'll be like, hey, adenine and guanine are examples of, and you're going to say purines, right? Okay, good. The phosphodiester bond. Okay, taking a look here at the phosphodiester bond. It's the reaction between one nucleotide and the other nucleotide. Now taking a look here, we can label these carbons on this sugar molecule. Okay, notice we have the nitrogenous base, and that can be one of 4. Okay, now taking a look here, all right, we can label the carbons. So 1, 2, that's already labeled. 3, okay, 4, and then here's the 5th one. Okay. Now the reaction occurs between the phosphate group of one nucleotide and between the 3 carbon, the hydroxyl group that's found on the 3 carbon of the next sugar that's made up of the nucleotide. Okay. Now this bond that's forming here is known as the phosphodiester bond. Now I might ask you guys to label the sugar molecule of a nucleotide. The reason being is because DNA, okay, is anti-parallel, which we will discuss. All right, and the reason why it's anti-parallel is because at one end of the chain it's 3, okay, that's the 3 carbon that's exposed. And when we see the other end of the chain, it's the 5 carbon that's exposed. So it's 3' to 5', or vice versa. What I want you guys to take a look at here, okay, this is all about DNA. So it's a deoxyribose sugar. All right, what does that mean? That means that it's missing an oxygen on the sugar molecule. It has a phosphate group, okay, as well as DNA consists of either A, T, C, or G. It's a double-stranded molecule, double helix. Okay, the DNA runs anti-parallel to each other. Notice the 3 carbon at one side goes this way, okay, to the 5 carbon of one of the nucleotides. The next strand over, okay, so the opposite strand is not going to be the 5' sugar, okay, or the 5' carbon of that sugar molecule. It's going to be the 3' that'll be at the end, and that goes this way. Okay, that's known as anti-parallel. One thing I do want you to know is the nitrogenous bases here. All right, what attaches these molecules together, okay, is not only the phosphodiester bond that occurs here between the 3' carbon, okay, and the phosphate group. What actually brings this molecule together and why it has its helical structure is the hydrogen bonds that occur between the nitrogenous bases A, T, and C and G. Now, how many hydrogen bonds occur between A and T? Well, 2. And how many bonds occur between C and G? 3. Does A pair with C and does A pair with G? No, it does not. A only pairs with T. Okay. Likewise, T only pairs with A. All right. So if I were to ask you, hey, what pairs with A? You would say T. And I'd say, hey, what pairs with T? You would say A. Now taking a look here, C. C only pairs with G. How many hydrogen bonds are between the two? 3. So if I were to say, what pairs with C? You would say G. And if I said, hey, what pairs with G? You would say C. Okay? This never changes. So A would never pair with G. A would never pair with C. If it did, there would be some issue within the DNA molecule, and it would be destructed in the cell. Or what could end up happening is you can get some crazy type of well, we know cell proliferation, right, or development of cancer if something like that happens, because that's known as a mutation. Complementary base pairing is the reason why A pairs with T and C pairs with G and vice versa. So T is a complement of A is the complement of T, okay, and so on. RNA, remember, consists of a ribose sugar. Okay, notice the hydroxyl group on this carbon. In DNA, it wasn't there. Okay, this was not there. That's why it's called a deoxyribose sugar. Okay, it also consists of a phosphate group, and the nitrogenous bases here, A, U, C, and G. Notice how there is no T. Okay, that's very, very important. It's single-stranded. All right, and essentially what RNA does is it converts information stored in DNA into proteins, and this is very essential when we discuss protein synthesis. Okay. Now I do want you to compare the two. I want you to compare DNA to RNA, and I want you to say that, hey, DNA is double-stranded. DNA consists of A, T, C, and G. Okay, DNA has a deoxyribose sugar. Okay, or you can say DNA is also hereditary information. RNA, okay, in this case here, RNA is single-stranded. It's made up of a ribose sugar instead of a deoxyribose sugar. All right, and it's the difference between the two is just the hydroxyl group that's found on the number 2 carbon. Okay, it's also consisted of A, U, C, and G. All right, and it's single-stranded. These are the macromolecules, guys. You got to get to know them because they are very, very important when we start to discuss cellular respiration, DNA replication, protein synthesis. They are very, very, very important. Okay, and I want you guys to review this as many times as you possibly can. Okay.